Now we'll be looking at another topic under form and function, which is respiration. And respiration is the chemical breakdown of glucose in the body to liberate energy for the activities of the body. Now, and in mammals, we're talking about mammals first. The respiratory organs of mammals begins with the trachea, then that's the windpipe. It moves to the glottis, which is protected against the entry of foreign particles by the epiglottis. Then immediately below the glottis, you have the larynx, which leads to the trachea. Then the trachea branches into two. We have like an inverted Y. That it looks like an inverted Y when you look at the diagram of the respiratory system. Then the trachea branches into two bronchi. The bronchi branches into bronchioles and eventually terminates into the thin wall of the alveoli, which are supplied with blood capillaries. Then the whole lung is covered by the pleural membrane and then the diaphragm. The diaphragm is at the lower part, that's the part that terminates the respiratory system. Then the lung is enclosed in a airtight compartment called the pleural cavity, or what we call the thoracic cavity. Now, that's just a brief intro or refreshing of your memory about what respiration is all about. Now to the questions. The first question we will be considering says, the gas that is produced during tissue respiration can be identified by using what? A. Sodium hydroxide, B. Calcium carbonate, C. Copper sulphate, D. Calcium hydroxide. In tissue respiration, looking at the various experiments you have to go through to identify or be able to detect which gas is being emitted and what happens when you use sodium hydroxide. The gas that you'll be identifying is oxygen. Oxygen, when there is a glowing sprint, when there is oxygen, it will keep on burning. Here we keep on burning when you have oxygen. So the gas that is produced during tissue respiration can be identified by using sodium hydroxide. That is our option A. Then when yeast respires anaerobically, it converts simple sugar to carbon 4 oxide and what? Option A says acid, B says water, C says oxygen, D says alcohol. The end product of anaerobic respiration, when glucose is broken down, you have carbon 4 oxide and alcohol. But if it is aerobic respiration, carbon 4 oxide and water will be given as byproduct. So that knocks off water, it knocks off oxygen and acid. So the alcohol alongside carbon 4 oxide are the end product of anaerobic respiration. The next question. Urea formation occurs where? A. Liver, B. Kidney, C. Heart, D. Lung. Urea is formed in the liver. By the activity of the liver, you have urea being formed as a byproduct. Tissue respiration is important for the absorption of oxygen in the alveoli, B. Release of carbon 4 oxide into the lungs, C. Release of energy for the body use, D. Exhalation of carbon 4 oxide from the lungs. I said earlier when I was trying to give a definition to what respiration is all about, I said is a breakdown of glucose into substances for the body to have energy to work with so that all the cellular activities can proceed. That's why respiration is very important. So that leads us to the option C, that tissue respiration is important for release of energy for the body. We take food, we take all sorts, but when it is broken down, then that is when there is energy that available for the body to use for other cellular activity. So that gives us option C. Tissue respiration is important for the release of energy for the body use. The next question is diagrammatic, showing a rubber sheet having thread, then we have balloon in it, analyzing the respiratory system. Now, we have the glass tubing, we have the bell jar. The experiment is inside the bell jar. We have the rubber sheet. We have 2 balloons. The 2 balloons represents the liver. The rubber sheets, that's at the lower part of the diagram, being represented by the diaphragm. Then the space in the bell jar is the pleural cavity. So, the question, the aim of the experiment is to demonstrate what? A, the presence of carbon 4 oxide in exhaled air. B, that in mammals, gaseous exchange takes place in the lungs. C, parts played by the pleural cavity and diaphragm in respiration. D, that a large amount of oxygen is absorbed by the lungs. Looking at the diagram properly, you see that the balloons, which represent the lungs, is deflated. It's not inflated. You don't find hair inside it, like it's relaxed. So, and then the diaphragm too, everything. So the aim of the experiment is to show that if the lungs is not available, then gaseous exchange does not take place. The lungs, represented by the balloon now, is deflated. So nothing will happen. No gaseous exchange will occur. So the answer to this question is that in mammals, gaseous exchange takes place in the lungs. Gaseous exchange takes place in the lung, option B. Then the next question, in the experimental setup, the rubber sheets represent the organ called what? The rubber sheets represent the organ called what? A, diaphragm. B, lungs. C, intercostal muscle. D, the pleural cavity. I have told you earlier what each of the components of the bell jar represented. That the rubber tubing it represents the trachea. Then the hair in the bell jar, represents the pleural cavity, that is hair tight. Then we have the rubber sheets being the diaphragm, the balloons being the lungs. So the answer to this question is A, diaphragm. The rubber sheets represent the organ called the diaphragm. The next question, the anaerobic fermentation of a glucose yields A, 38 ATP molecules, B, 2 ATP molecules and alcohol, C, pyruvic acid and alcohol, D, water and carbon 4 oxide. In anaerobic fermentation, the glucose molecule will yield pyruvic acid and alcohol. Pyruvic alcohol is yielded, and we have pyruvic acid in anaerobic fermentation. That is option C. The next question, the opening of the stoma is controlled by what? That is in leaves of plants. We have in the stomata, we have the gaseous stoma, or in the leaf of plant, and that is the organ of respiration, the opening and closing of the stomata. Now, the opening of the stoma is controlled by what? What controls the opening and the closing of the stomata? A, presence of a pore. B, increase in solute concentration in the gas cells. C, presence of gas cells. D, decrease in solute concentration in the gas cells. When the solute concentration in the gas cell is increased, then the stoma will open so that some can be removed. But once there is decrease, that is, it's not even enough for the plant, you find that the stoma will be closed. If we have to manage what is available, that is why you find reduced leaves, and even the stomata in plants in areas where there is reduced or little or no water in the environment. You find that their stomata is very reduced, so they don't have to be losing that water by transpiration every time. So once there is an increase in the solute concentration, like you have more than enough, you have excess, there is excess, what the plant needs, then some that you can afford to just get to be taken away from the plant, then the stomach will be open. So that leads us to the option B, increase in solute concentration in the gas cell. Once there is increase in the solute concentration, fine, the stomach will be open. But when there is decrease, that's option D, decrease in solute concentration will lead to the stomach closing. Then, so that's the answer to this question now. The opening of stomach is controlled by the increase, option B, increase in solute concentration in the guard cells. The next question. The mammalian lung is made airtight by the A, the pleural cavity, B, the mucous membrane, C, the pleural membrane, D, the diaphragm. I have explained the respiratory system, the components earlier, but because of this question, I'll go over that again. That the diaphragm is at the lowest part of the respiratory system, and that is contract and relax during inhalation and exhalation. Then the pleural membrane is internal to the respiratory system, is inward. So when there is respiration is ongoing, the pleural cavity is there. The mucous membrane is just what is responsible for moistening of hair. When you breathe in hair in your nostrils, the mucous membrane is there, it moistens the hair